Mark, just give me a second. Students have joined for the biology lab. There is some confusion regarding the link.
Okay.
Hello, am I audible, students?
Yes, ma'am.
Yes,
All right, good evening, everyone. How are you all?
good
Are you
ma'am.
doing
How are
good?
you?
Fine, ma'am. How are you?
I'm also good. All right. So, did you people have any difficulty in the previous topic that we had discussed?
No,
No,
ma'am.
ma'am.
What is the last thing that we had discussed? Was it sickle cell anemia or hemophilia?
Ma'am, it was hemophilia.
Hemophilia. Okay. Okay, students, can you see the screen?
Yes,
Yes,
ma'am.
ma'am.
All right. Okay, students, switch on your video. Let's start today's class.
Yeah, ma'am. Just a minute.
Sure. Take your time. Sadhika, your mic is fixed now?
Yes, ma'am.
So glad. Finally, I can hear your voice. I think I remember, like, I have done this thing with you all, or I think I have done sickle cell anemia, like the shape of the RBCs were changing to sickle. Like, do you people remember this? Like, I have a flashback as to I have discussed this with you. Anyway, we'll discuss it again. Yes, everyone, switch on your videos quickly. Let's start today's class. I don't know why many students have not joined today. Hemophilia was done. So let's discuss sickle cell anemia yet again. Now, see, sickle cell anemia, it is represented by the gene S. Now, for all those human beings that have HbA and HbA, those are normal homozygous humans. The ones that have HbA and HbS, now they are normal, they do not have the disease, but they are the carriers of this particular disease. And then HbS, these are the ones that are disease. And these people that have sickle cell anemia, they die before attaining proper maturity in their lives. Now, this HbS is the mutated gene. Now, what happens in this particular case? There is a proper transversion mutation that happens in the beta chain of hemoglobin. Now, you people might remember that I said that RBCs, they are red in color because they contain hemoglobin, right? So those hemoglobin that is present in the RBC, they are actually made up of two chains. You can say alpha and beta chains. So this error or this mutation arises in the beta chain of the hemoglobin. So because of the transversion mutation, so what happens here in the beta chain of the hemoglobin, because of this particular mutation, initially at the sixth position of the beta globin chain, there is glutamic acid. But in this mutation, this glutamic acid gets replaced by valine. And glutamic acid or valine, both of them are the examples of what, students? What are they? Glutamic acid and valine. What are they? They are amino acids. So one amino acid is getting replaced by another amino acid, and where? So at the sixth position of beta globin chain. Now the mutant hemoglobin. Now the hemoglobin is a mutant hemoglobin. It is not the normal hemoglobin. So it undergoes polymerization. It starts forming polymers under low oxygen, and it changes the shape of RBC. Now normal shape of RBC is biconcave disc shape. But from this particular shape, it becomes elongated sickle-like structure, which reduces the oxygen carrying capacity in this particular RBC. So this is what happens in the sickle cell anemia. So now you people have understood like which chain of hemoglobin gets affected, alpha or beta? Which one is it affecting? This mutation? It is affecting the beta chain, right? Where are the rest of the students? Why are you not switching on your videos? Khadija, Mariam, Ikra, what happened? All right, so the next disease that we are going to talk about is phenylketonuria. Now, phenylketonuria is not a sex-linked disease. Rather, it is an autosomal recessive disorder. Again, for the expression of this particular disease also, there has to be the recessive homozygous condition. Now, phenylketonuria, it is an error of metabolism. Why? Because there is an enzyme that is phenylalanine hydroxylase enzyme. This enzyme is not prepared or not made by the body in the disease, that is phenylketonuria. So, the individual affected by phenylketonuria, they lack the enzyme that is known as phenylalanine hydroxylase. Now, what is the function of this enzyme and why is this enzyme important? See, in our body, under normal conditions, what happens is that this phenylalanine hydroxylase enzyme, it converts the amino acid phenylalanine into the amino acid tyrosine. This is like normal metabolism that happens in the body. And since this is not present, like this phenylalanine is not present, so because of the absence of phenylalanine hydroxylase enzyme, the amino acid that is phenylalanine, it will not get converted into tyrosine and it will start getting accumulated in the body. So as a result, accumulation of phenylalanine takes place, and because of that, it can, like, a lot of phenylalanine together gets converted into phenyl pyruvic acid. And the accumulation of these, it causes the mental retardation. So this is phenylalanine. Now, phenylketonuria is another example of which type of inheritance, students? We are talking about it in the terms of Mendelian disorders, but what else have we studied about phenylketonuria? Do you people remember? Anyone? Do you people remember pleiotropy? No one is responding. You say yes or no. What is happening? Where are you, students?
Yes, ma'am.
You remember pleiotropy?
Yes, ma'am.
So it is example of that also. Now moving forward, we'll talk about the next disease, that is thalassemia. Now thalassemia is autosomal recessive disorder which is caused by mutation, or you can say the deletion kind of mutation. And in this, defect in the synthesis of globin chain of RBC takes place. So see, in the hemoglobin, there is a heme portion and then there is a globin portion. The globin portion is the protein and the heme is the iron compound portion. So here, the globin part of the hemoglobin is not properly formed. It is malformed. So because of this, thalassemia takes place. Now this thalassemia, it is of two types. There is alpha thalassemia and then there is beta thalassemia. In the case of, and do you remember I said that hemoglobin is made up of two chains, that is alpha and beta chains. So in the alpha thalassemia, the production of alpha globin is affected. The alpha globin is not made properly. Whereas in beta thalassemia, the production of beta globin chain is affected. Depending upon the name, beta chain will be getting affected. In alpha thalassemia, the alpha chain will be getting affected. Now remember here, the names of the genes are important to remember. See, HBA1 and HBA2 genes present on the chromosome number 16. They are responsible for the production of normal alpha hemoglobin. And then in this case, HBB gene on chromosome 11, it controls beta thalassemia. Any doubts up until now?
No, ma'am.
Okay. And then after this, there is like a sort of like a comparative analysis between thalassemia and sickle cell anemia. Now talking about thalassemia, so it is a quantitative problem where too few globin molecules are synthesized in this. But in the case of sickle cell anemia, it is a qualitative problem because here the number of globin chain is not important. Here, the quality of the globin chain is important. So here, sickle cell anemia is a qualitative problem, whereas thalassemia is a quantitative problem. Less number of globin molecules are synthesized. Any doubts up until now? Any doubts? Also, what is the last thing you people have noted down in your notebooks? Can you see the last line so that I can give you time to write down from there only? What is the last thing? Yes, tell me what is the last thing that you people note down in the previous class, the last class that we had?
Ma'am, it was hemophilic female dies before birth.
Okay. So now you people have to note down from here. All right, now you can note it down. You can switch off your videos and note down.
Ma'am, done.
Everyone's done?
Ma'am, done. Ma'am, done. Ma'am, done.
Everyone's done?
Ma'am, done. Ma'am, done. Ma'am, done. Ma'am, done. Ma'am, done. Ma'am, done. Done, ma'am.
All right. Any doubts up until now?
No, ma'am.
Now, the next thing that we are going to discuss is the genomatic mutation. Now, genomatic mutation is basically the number kind of mutation, meaning the mutation because of which there will be a change, or you can say the variation in the number of the chromosomes. And this brings the effect on the phenotype as well. So, talking about the genomatic mutation, it is the change in the chromosome number that brings about visible effects on the phenotype of the organism. Now, this genomatic mutation, it is of two types. It is aneuploidy, which contains monosomy and trisomy. And then it is euploidy, that is haploidy, only one set of chromosome, polyploidy, more than two sets of chromosome. See, the normal chromosome is 2n, which is diploid condition, meaning 46 chromosomes are there. In the case of monosomy, there is 2n minus 1 condition, meaning 45 chromosomes, which is aneuploidy. Then when 2n minus 2, meaning out of 46, you remove 2 of the chromosome, then that is nullisomy. And then 2n plus 1 is trisomy, meaning 47 chromosomes in totality. So here the number of chromosomes are increasing or decreasing, and that is what we call as aneuploidy. Now, in this case, the euploidy, it is how many sets of chromosomes are increasing. Here only single, or the number of chromosome is like usually 1 or 2. But in the case of euploidy, it is basically the sets of chromosome is concerned. So talking about the haploidy, so in the case of haploidy, there is only one set of chromosome, meaning N. Meaning out of 46, there is only 23 present. But polyploidy is more than two sets of chromosome. You have like either double of the 46 chromosome or triple of it. So that is euploidy, meaning here the sets of chromosome increase or decrease. But in the aneuploidy, the number of chromosomes increase or decrease. Now, the next thing that we are going to discuss, we had already discussed the Mendelian disorders, and now we'll be discussing the chromosomal disorders. Now, see, chromosomal disorders are the ones that are caused due to imbalance in the chromosome number. In that case, there was the proper genotypic structure and everything that was concerned. But here, now what happens is that the imbalance in the chromosome number is there, because of which a lot of problem arises in the body. The first case you like all of you might have heard about is Down syndrome. Now, Down syndrome is one of the most, you can say, common type of chromosomal disorder that is like found in these days, and it was first described by Langdon Down in the year 1866. So on his name only the name of the syndrome is kept like that. And this disease is occurring due to the trisomy of chromosome number 21, meaning trisomy is what? Trisomy is 2n plus 1, meaning apart from 46 chromosomes, there is an extra chromosome making it 47 chromosomes in totality. So now what happens in the case of Down syndrome? So the person that has Down syndrome is short statured, meaning has a short height, has a small round head, has a furrowed tongue, and broad palm with palm creases present in all those people that have Down syndrome. Then there is like the tongue is also wrinkled, and the physical, psychomotor, and mental ability along, meaning physical means gonads, meaning the testes in the case of male and the ovaries in the case of females, and also their genitals. They are like, you can say, retarded, meaning the development is not proper, like the poor development of them is there. Then the next disease is, now the Down syndrome is clear to everyone.
Yes, ma'am.
Any doubts in Down syndrome? Why is
No,
it
ma'am.
happening? What is the case? Trisomy is also clear to everyone.
Yes, ma'am.
All right, very good. Moving forward, the next disease that is a chromosomal disorder is Klinefelter syndrome. Now, Klinefelter syndrome, it happens in the case of males. And what happens here? An additional X chromosome is present, meaning normally a male has XY, but in the case of Klinefelter, the male has XXY. One extra X chromosome is there. So, in totality, there is 44 plus XXY chromosome set. Now here, why does this kind of disease can is happening and what is the reason behind this? So this defect occurs due to the union when an abnormal egg and a normal sperm fuses, meaning an egg which is 22 plus XX, meaning has an extra X chromosome, and the normal sperm that has either the Y or the X. And apart from this, the opposite condition can be the egg is normal but the sperm is abnormal. So such persons, they are sterile males, like they cannot reproduce. They look like males, but they have some female characteristics in them because of the presence of an extra X chromosome. Now, what are the female characteristics that are there? There is feminine pitched voice, then the development of breast, which is known as gynecomastia. So even though these persons that have the Klinefelter syndrome, these males, they are males only, like they are male, but they have certain female characteristics in them, which because it is because of the extra X chromosome. And some of the female characteristic is voice like a female, and also the development of breast in male. It won't be functioning breast, but it will be the development of breast in the case of males. That is known as gynecomastia. Any doubts in Klinefelter syndrome?
No, ma'am.
All right. Now the next is Turner syndrome. Now Turner syndrome takes place in the case of females, and here it happens because of monosomy. Now monosomy is an example of aneuploidy, meaning either one or two chromosomes can be missing or can be extra. Now here what happens? It occurs due to the fusion of abnormal egg, meaning the egg that has the body chromosomes, but it does not have the sex chromosome, meaning 0, or and the normal sperm. So in this case of fusion, the Turner syndrome can take place, or the next condition is the egg is normal but the sperm is abnormal. So in both of these conditions, Turner syndrome will take place. Now talking about these females, they are also sterile. Why? Because their ovaries are rudimentary. Rudimentary means they are poorly developed. They are not proper, like there is no development in the case of ovaries. And apart from that, these females, they also lack secondary sexual characters, meaning those characters that are responsible for forming a female like appearance, the like all of these secondary sexual characters that are responsible with puberty and everything, they do not have it. Okay, so this is why Turner syndrome is also considered as a chromosomal disorder, because here only because of the presence of an extra chromosome, or because of an the absence of an extra chromosome, this disease is arising. So this was about Turner syndrome. Any doubts?
No, ma'am.
All right. Now you people can note down till here, and then we'll talk about the next topic.
Ma'am, done. Ma'am, can you please minimize the screen?
I'm not able to minimize there. It's like not I'm trying to, but it's not happening actually. You can copy down.
Okay, ma'am. Ma'am, done. Ma'am, done. I'm done. I'm done. I'm done. I'm done. I'm done. Ma'am, done. Ma'am, done. Ma'am, done.
All right. Any doubts up until now?
No, ma'am.
All right. The next topic is pedigree analysis. Probably this is the last topic of this particular chapter, and there are like nothing else that is left in this particular chapter. So, all right. So, talking about pedigree analysis, let me just give you a quick overview of this because we'll do it in depth in the next class. But this is the analysis through which you can study how the genetic disorders are being inherited in the humans. And this can be done over several generations. So, talking about the pedigree analysis, it is the it is the method of study of human genetic disorders. The analysis of traits in several generations of a family is known as pedigree analysis. Now here what happens, we just like how in younger classes we used to draw family trees with pictures. This is the family tree only, but with the genotypes and like how the inheritance took place. Now we know that what are the Mendelian disorders, and this pedigree analysis can actually be used to find out the